Hey everybody, welcome to another video lecture, this one on chapter 12, where we look at protein synthesis, which is, we did transcription last time. This is translation of the genetic code. And we're basically going to go through all of these. We'll spend probably the least amount of time talking about eukaryotic translation. We're going to learn everything about translation looking at prokaryotes, and then look at some differences as far as eukaryotes are concerned. So we'll come back to that. All right. So protein synthesis is going to require a few components. We're going to need ribosomes. These are the sites at which protein synthesis actually takes place. We're going to need that messenger RNA that was transcribed in the last chapter that we looked at. And then we're going to need transfer RNAs, these tRNAs. And all of these things are going to kind of complex together. We're going to see that tRNAs bring in the amino acids necessary for the growing chain of the peptide. And then there's going to be some protein components. We'll call them initiation factors. And we'll look at those a little more closely when we get to chain initiation, elongation, and termination. So we're going to need to activate the amino acids. That's going to be the first thing we sort of spend time looking at before we get into these guys. We're going to look at how these things get, these amino acids are activated, and then after that, before that really, we're going to look at just the nature of the genetic code. So we're going to come back to the amino acids and how they get activated. tRNA and a special enzyme are involved. Chain elongation will repeat over and over. And similar to transcription, how transcription had initiation, elongation, and termination, we're going to follow that same sort of theme. So our genetic code is a triplet code, and we know this. We're going to go through some of the experiments on how we know this, but since we do know it, we can say that at this point. We have a triplet code, and every 3 bases is what we're going to call the codon, and codons specify specific amino acids. There are 20 amino acids that are commonly found in proteins. There are also amino acids that are not found in proteins, that are in our bodies, though. And again, to be an amino acid, you're just you have an amino group on the alpha carbon of carboxylic acid. So amino acid is just a general term. We just happen to have 20 common ones, a couple less common ones, and then some rare ones involved in proteins, and then of course others that are not involved in proteins at all. Each amino acid can have more than one codon, but no codon can encode more than one amino acid. And you'll see the code here in a second. It's non-overlapping, so we have a non-overlapping code. Here you can see the difference. We know that we have a non-overlapping code because a strand of mRNA only yields one product. If we had an overlapping code, then you can imagine that the same little stretch of mRNA could actually give multiple products. And here it's only shown 5 different sort of ways of reading this. But we could read these first 3 as a code, or we could read these 3 as a code, or we could read these 3. Oops, it's the same. No, those 3 as a code. So you can see that different ways. And then from there, of course, every 3 after that. And so you could transcribe a piece of mRNA many different ways and get lots of different proteins, and we don't see that. So we know we have a non-overlapping code. We also know that it's continuous, meaning that there are no gaps in between the codons. And so a punctuated code illustrates what that might look like if there was gaps. So we say our genetic code is commaless. Now there are 64 codons that can occur with 3 base pairs, right? If you look at 3 nucleotides and how many different ways that you can take those 4 nucleotides and arrange them, there's 64 different triplets. This is necessary. because if the code was a single digit, like 1 to 1, then like if an A coded for, let's say, alanine, and then a G coded for glycine, and so on, well then obviously we would only have 4 amino acids, right? We would only get 4 amino acids. If you use a double code, so let's say AA for alanine, GG for glycine, CC for whatever, cysteine. That's not how you do cysteine. Anyways, you guys know what I'm getting at. If you use double codes, well, that only gives you 16 possibilities. You still don't have enough codes to get all the 20 amino acids. So nature necessarily needed to have a codon be 3 nucleotides so that we could have enough possibilities to encode 20 amino acids. Now, because there are so many extra codons, we're going to see that some actually code for multiple of the same amino acid. Of the 64 codons that we can get by arranging those 4 nucleotides in different ways, 61 of them actually give us amino acids, while 3 of them are going to be stop signals. And all that really means is that there aren't going to be tRNAs that recognize these, and no tRNAs means no amino acid, and so these will be our naturally occurring stop signals for the growing peptide. All right, so here you can take a look at the genetic code. So you can see all the 64 different codons and what the 61 that code for amino acids, which amino acids they code for. What you'll see here is that the first 2 nucleotides in the codon pretty much determine what amino acid you're going to get. And then the third position, we can call it the wobble position because it kind of fluctuates. It doesn't really matter what it is in some cases. So like what I mean is in 8 cases, in 8 families of amino acid, the third nucleotide doesn't change the amino acid. So all of those in gray, you can see that. So let's look at this one. Starting letter U, second letter C, third letter can be a U, a C, an A, or a G. In any case, we're going to get serine. And so that's the case for 8 of those amino acids. There are some other situations here where if you have a purine in the third position or a pyrimidine in the third position, you actually get a different amino acid. So you can see that one. I guess this one right here is a good example. So first letter U, second letter U. If we get a pyrimidine in the third position, we get phenylalanine. If we get a purine in the third position, we get leucine. There's one situation where 3 out of the 4 possibilities in the third position will code for isoleucine, and the fourth position we get actually your book will define it as 2 unique codes. So AUG only gives you methionine, and UGG only gives you tryptophan. So this is where the third position has to be a G, can only be a G. And then there's one, the stop codon here, where the third position can only be an A. But you'll see that third position, it is variable. We say that the genetic code is degenerate. That means that more than one triplet code for the same amino acid. There are it's multiple tRNAs giving you the same amino acid. This is good for a few reasons. As you saw before, 4 amino acids, 4 tRNAs or 4 codons, all giving you phenylalanine, means that if there's a mistake with transcription, and that third position accidentally gets, substituted a different base in there, we end up with the same amino acid. So no harm, no foul. So it's good to have that. One of the other things about the degenerate code is that you don't have to have lots of one particular tRNA, right? To code for phenylalanine, we don't need to have tons of the phenylalanine tRNA. We actually can just have normal levels of those 4 different tRNAs, and phenylalanine gets kind of coded for anyways. So having more than one tRNA that codes for the same amino acid is a good thing. Now, the genetic code is generally thought to be, or said to be, universal. We see the same codons code for certain amino acids in almost all life. Viruses, bacteria, animals, plants, us. There are exceptions. Rare organisms have sometimes some, and I don't mean they're rare because there's anything special about them. They're rare in that they have exceptions to this rule. Mitochondria are included in this. Sometimes their stop code or the normal stop codon that we would have actually codes for something like glycine in these organisms, or something that codes for serine might actually be a stop codon. So there are a few exceptions, but for the most part, this genetic code must have evolved before all the different, organisms that have kind of come after it. How did we figure out what the genetic code is? So there was a couple of experiments, and one of them was just to figure out, hey, what kind of code do we have? Is it a single digit code? Is it a double digit code? Is it a triple digit code? They figured this out. And is it a continuous reading frame, or are there multiple reading frames, or as we were talking about earlier, continuous versus discontinuous, and punctuated or not. And so the experiment that they used was pretty neat. What they did is they synthesized some artificial mRNAs. So let's say they did one with a bunch of U's. It's a poly-U mRNA. And the only codon you can get from a poly-U mRNA, right, is UUU, UUU, UUU. And so this thing would obviously code for, well, it does. It codes for phenylalanine. We could go and look at that table, and we would see phenylalanine. But it codes for, what does your book call it? Homopolypeptide. So the polypeptide and that multiple amino acids linked together, but they're all the same. So this is just a big long chain of phenylalanines. And so then they did that. They figured out phenylalanine must be something to do with U's. And so then they went and did a bunch of A's, right? And then again, A can only code for one thing. And so then they got a polypeptide that has only one amino acid. Okay, so then they have that. So they figured out, UAGC. They did all those. Then they switched into doing some double codes, some alternating sequences. And so then they would draw something like this, or they'd go like AU, 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 and then see what comes out of that. And so if you see this, AU, AU, it's actually an alternating code, right? AUA, and then UAU, and then AUA, and then UAU. So what you end up with in terms of the polypeptide is that you get an alternating sequence of amino acids. Okay? And so this was part of the experiment that also helped them figure out that it was a triplet code. Let me kind of explain how that happened. So not only is it a triplet code, but they figured out that there was not multiple reading frames, that there was only one reading frame. Okay, so kind of follow this. See if you can follow this. So let's imagine that they did this. AU, 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 AU. This is what they get. They get an alternating polypeptide. Okay, well what if it was a double code, a two-digit code? Not a triplet, but a doublet or a duet. Well, let's kind of look and see what we would get. Well, we would get AU, 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 AU. We would get another homopolypeptide, right? It would be a polypeptide that has the same nucleotide. The same amino acid over and over and over. So this is, again, how we know two things, that it's not a doublet, but also that it's not continuous. What if the reading frame wasn't AU, but it was UA? Because at this point, remember, they didn't know about the start codon, or they were figuring all this stuff out. So they didn't really know where translation was going to start on this chain. It could have started with AU, but it could have also started with UA. But in either case, if it was AU, we would get a polypeptide with all the same amino acid. If it was UA, we would again get a polypeptide with the same same one amino acid over and over and over. If it was both, let's say that some ribosomes did it one way and some ribosomes did it another way, then we would actually get a half and half mixture of two polypeptides, but both those polypeptides would be made up of only one type of amino acid, right? And none of those things happen. We don't get homopolypeptides. We also don't get two kinds of homopolypeptide. We only get one kind of polypeptide and its alternating amino acid sequence. And that's not what we would get if it was a doublet or if there was continuous reading frames. So that kind of makes it so that the only possibility is these things are triplet codes and there's only one reading frame. There's no other reading frames because a different reading frame would give us UAU, AUA, UAU, AUA, as opposed to, well, of course, in this one, I don't, with a doublet, with a repeating double sequence, we're not going to get much variation, so bad example, but we would get different polypeptides. Okay. How do we figure out what the actual 3-digit codes are for all of the amino acids? So this was done, I'm actually going to just jump over to this slide here. What happens is they were able to affix a ribosome to some filter paper that had an artificial anticodon generated on it, so a CAU. So we had CAU, we associate it with a ribosome so that a tRNA will bind, and then we carbon 14 labeled one of the mixture of tRNAs. And we want to see, which tRNA is actually going to bind to the anticodon CAU. And so in this first example here, in first A, they've tagged a proline. Well, proline has the anticodon AGG. Sorry, this is the anticodon here. This is the codon, CAU. So the artificial one is CAU. Whichever tRNA has the anticodon that matches that will pair here. And so they label the proline to see if proline possibly matches CAU. In this experiment, it doesn't. The proline doesn't bind, and so it just passes through the filter paper, and then they don't detect C14 on the filter paper. Nope, not there. So then they know that CAU is not for proline. So then one by one, they go through that. And eventually, they will get something that binds. And so in this experiment, histidine is the one that was tagged. And then histidine actually has the anticodon on that tRNA that matches the codon CAU. So histidine would bind to this ribosome, and then they would detect the radioactivity on the filter. And that's how they figured out which amino acids have which codes. And they did this 64 times. All right, so the codon will form a base pair with an anticodon on the tRNA so that the tRNAs are recognized and bring in the proper amino acids. So the anticodon is the sequence on the tRNA. Some tRNAs bond to one codon exclusively, but other tRNAs, or many, can actually recognize more than one codon because of something called the wobble. And this has to do with different hydrogen bonding patterns. And again, we looked at this when we saw that there are 4 different bases that can be in the wobble position. The third position is the wobble position in our codon, but it's the first position in the anticodon. And again, having multiple bases that will still recognize the same codon helps us. It's a better mechanism for fighting against mutations that can happen. So I'm going to show you a couple of things here. First, just what that bonding looks like. So here's the codon on the mRNA. Here's the anticodon on the tRNA. The wobble base is that third position here. But remember, we read DNA from 5' to 3'. So if we were reading the tRNA's anticodon, we would read it as GGC. Okay, let's look at how some of those other bases pair. Now, there's a base called Hypo Xanthine that we see a lot in the 5' end of the anticodon, so the first position of the anticodon. This one can form some hydrogen bonding with A, C, and U, which is a little more unusual than what we're used to. So let's take a look at those. So normally we would see A pairing with T, the purine pairing with a pyrimidine. Here we see actually AG pairing up, so we can see that there are some hydrogen bonding that can happen. We see UC pairing up. Here we see that hypoxanthine pairing up with an A. Uracil can form two types of hydrogen bonding. You can see this just kind of shifting it down, and then it's down here on these two as opposed to these two. Anyways, so we have some hydrogen bonding that is still able to happen. The only time we don't see any weird hydrogen bonding is with an A or a C. Whenever there's an A or a C in that first position on the anticodon, A always pairs with U and C always pairs with G. But when it's one of these guys, it's a little more flexible. So, yeah, we talked about this a little bit. Fewer different tRNAs are needed because a given tRNA can actually base pair with a few different codons. And then the existence of the wobble means that by any mistakes that happen during transcription don't necessarily lead to a different amino acid. Okay, let's talk about the reactions that take place to activate the amino acid. So, excuse me. Both of these steps, the activation steps, are both catalyzed by the same enzyme, tRNA synthetase. Step 1, we're going to have the amino acid actually become enzyme bound, and then during step 2, the enzyme bound amino acid is going to get attached to the tRNA. So let's take a look at that. We'll talk about this. Okay, so here's the diagram. So A and B. A is just showing us the overall reaction, that an amino acid and a tRNA with some energy is going to form an aminoacyl tRNA, so a loaded up tRNA, an activated amino acid. B here is showing us those two steps. So step 1, we have the enzyme, ATP, and the amino acid. We're going to get some energy. We're going to hydrolyze that ATP bond. We're going to get our enzyme, that's enzyme bound now to this amino acid, and we're going to have transferred an adenine monophosphate onto that molecule. So this is a enzyme bound aminoacyl adenylate, or an aminoacyl AMP. So now this thing is ready for the next reaction, which is going to be connecting it to a tRNA. Now, there are two classes of aminoacyl tRNA synthetases. They think this is actually just convergent evolution, two different times, this evolved twice. Depending on if it's class 1 or class 2, we're going to get a bond forming on the 2' ribose or the 3' ribose of this group that's attached to the tRNA. So the tRNA also has an adenine monophosphate on it, and that's where we're going to form the bond. And so depending on if it's class 1 or class 2, we're going to get a 2' aminoacyl tRNA or a 3' aminoacyl tRNA. Okay, it kind of looks like from the book here that the 2' ultimately get turned into the 3'. So this is the activation of the amino acid. So loading it onto the the tRNA. Now the tRNA has, the amino acid tRNA actually does some double jobs here. In addition to synthesizing this or catalyzing this reaction, it's also proofreading. So the two proofreading sort of activities is one, it has to recognize the amino acid, the correct amino acid. So every amino acid has its own tRNA, and every tRNA has its own amino acid synthetase. Is that the name of it? Amino acid tRNA synthetase. So if there are, I don't know how many tRNAs, every tRNA has its own synthetase. So the synthetase has to recognize the amino acid and recognize the tRNA. If it doesn't recognize both, then this reaction doesn't happen. So this is kind of a proofreading mechanism to make sure that no tRNAs are accidentally bringing in incorrect amino acids. The tRNA, once it's bound, so what we're looking at here is the specific binding sites that are recognized on the synthetase, on the enzyme, or the specific binding sites on the tRNA that the enzyme recognizes. And so you can see here for these amino acids, it recognizes this sort of variable pocket. For these amino acids, it's the acceptor stem that it's recognizing. These amino acids here, it's also recognizing position 37, or 73 rather. Obviously the anticodons are important recognition points. For these two, it's this variable stem loop here. But again, they don't have to be the anticodon, and they can also be different parts of the tRNA molecule that the enzyme is recognizing. So there's going to be some variations, obviously, within the different tRNAs. Okay, so we talked about this. Chain initiation, chain elongation, and chain termination are going to be, again, similar with transcription, the stages in translation. And what we can see here is actually an X-ray crystallography model or structure of a ribosome, shown in blue, with a tRNA here in orange. So this tRNA, this is in gold, I believe, this is the mRNA that it's locking onto and kind of reading. We don't see the amino acid that I can tell in this picture, and I'm forgetting right now what this big blob is. It's not the synthetase, it's something else. Okay, Peptidyl Transferase is the name of this big protein. This protein, we'll see, is actually responsible for catalyzing that peptide bond between the amino acids. Okay, so chain initiation is the beginning, obviously, of this process, and we're going to look at sort of the parts that have to be there. Now, the chain, we know, grows from the N side to the C side. This is why we read from N to C and why we read the DNA from 5 to 3. So in prokaryotes, the first amino acid that always gets incorporated is N-formyl methionine, or FMET as it's abbreviated. So this will always be the start codon. Now N-formyl methionine is made from methionine. It does have its own tRNA, but the methionine that's bound to the FMET tRNA gets formylated. So it gets this group added to the nitrogen side. So it actually does have its own tRNA, but both of those tRNAs are originally loaded up with methionine. The one that's meant to be formulated has something different on it that sort of lets your body know that reaction needs to happen. So this is the conversion of methionine into N-formyl methionine, that added group there. And again, their specific sequence is going to be AUG. This is the start codon in the mRNA. And the tRNAs are going to have that anticodon UAC. that pairs with that. We're going to look at a diagram here, and it should be a little easier to look at. So when these RNAs recognize the start signal, this is going to be the beginning of the synthesis. Now there's going to be some other components that are required. So in addition to there being obviously mRNA and that start amino acid in tRNA, there has to be all the other amino acids in tRNAs also present. We're going to need ribosomal subunits. You're going to see that there's the 30S and the 50S, and they will combine together to make the 70S. We're going to need initiation factors. These are proteins, the proteins that we talked about right at the beginning of the chapter, the protein components. Initiation factor 1, 2, and 3. We'll talk about what those do. We're going to need GTP. GTP is the main source of phosphorylation in this, or phosphate groups. So we're going to hydrolyze GTP into GDP for a lot of the steps and transfer that phosphate group to our tRNAs, or sometimes to components of the protein components of this big complex. Okay, so yeah, we're going to need all these pieces. mRNA, ribosome, proteins, amino acids, initiation factor proteins, amino acids, and then energy in the form of GTP. So, okay, so what's shown here is sort of the, now you're wondering, okay, so when all of these things are around, what happens? Well, the initiation complex is going to require a few things. Those initiation factors are going to help recruit the 30S ribosomal subunit to the mRNA. The mRNA has on it, we'll look at that in a few slides, something called the Shine-Dalgarno sequence that's going to help position the ribosome in the right spots so that we're going to see that there's some sites here in the ribosome. This is the P site, so that the P site lines up right on the start codon. Our tRNAs are going to come in. The tRNA here is associated with the initiation factor 2. We're going to name these initiation factors here in a second. Let's see, what else? Okay, so once this is bound, we need to then recruit the 50S subunit, and so that's what's going to make the entire initiation complex. All right. IF3 helps to recruit the 30S subunit and hold it on the mRNA, but it also prevents the premature binding of the 50S subunit. IF2 actually binds GTP and aids in the selection of that initiator tRNA. And so it's part of the mechanism to make sure that the correct starting tRNA goes in there. And then the IF1, there's not a whole lot actually known about IF1. It's thought to just kind of hold IF3 and IF2 kind of in place, and also kind of facilitates both of their jobs. So it helps IF3 keep the 50S subunit from binding prematurely. It also helps IF2 with the selection process of that initial tRNA. Now, the Shine-Dalgarno sequence is a purine-rich leader sequence that matches a pyrimidine-rich sequence in the ribosome. It's about 10 nucleotides upstream of that start codon. And again, it'll recognize part of the ribosome, and it's there to help align properly. We're going to see something similar in eukaryotes. Here are a couple of Shine-Dalgarno sequences from various organisms. You can see that it's relatively conserved. There's a whole, in either case, there's a lot of A's and G's, purine rich. And then here we see the sequence in the ribosome, part of that ribosome that actually recognizes this. And so again, pyrimidine rich. All right, chain elongation. This is where we're actually going to form peptide bonds. The ribosome's going to move along the mRNA or pull the mRNA through it. and we're going to basically grow the peptide. So there's a few sites that are important here. We're going to call them the E site, the peptidyl site, and the amino acid site. So E, P, and A. So I'll just show you the diagram. Where's that diagram? So here it is. Now we can talk about it and then we can go back and look at all the stuff I skipped. So here we have our 70S subunit all bound, ready to go. So we're going to need some elongation factors that come in here, and there's an elongation protein also that comes in. So this EFP is going to come in here. The job of EFP is thought to help with that initial peptide bond, so the forming of the peptide bond between F-met and alanine. We're also going to need some other elongation factors. There's elongation factor TU, elongation factor TS, in addition to that elongation factor protein. TU, its job is actually to bind the amino acid tRNA. Once we've bound the amino acid tRNA, we're going to hydrolyze a GTP. This is going to help facilitate getting that amino acid into the A site. So the A site is the neighboring site to where we've already got that P. Once EFU has helped the A site get filled, and EFTU is actually one of the most prominent proteins in the cell. It's a very, very important protein. It's like 5% of the dry weight of the cell in these prokaryotes. This guy here, its job is to make sure that the correct tRNA gets in there. If the binding isn't right, this process stalls out. If it's too loose or too tight, it doesn't work. So EFTU's job is to check that amino acid actually matches that anticodon. The anticodon on the tRNA actually matches that codon, and then it puts it in there. It hydrolyzes the GTP and it dissociates. So then it falls off. The job of EFTS is basically just to regenerate EFTU. Okay, so then we've got a new amino acid tRNA in the A site. So now we're here. All right, so then peptidyl transfer happens. So EFP, catalyzes the transfer of this amino acid chain, or in this case it's just one amino acid, but as this gets longer, the chain will get transferred over via nucleophilic attack of the nitrogen on the C-terminal end of that methionine, nitrogen from the alanine, C-terminal end of the methionine, and we get a transfer. And so you'll see that right now N-formal or formal F-met is on the P site, and then down here F-met now you see is in the A site. So just moved over to the A site. So now our peptide chain is getting longer. The P site now has an empty tRNA, and what's going to happen is we're going to need to move everything over. So this elongation factor G comes in. This thing again will use GTP energy to basically move these guys over. So the uncharged TNA sort of falls off. This is just going to kind of fall off. But the peptidyl tRNA needs to get moved over to the P site. And so it gets translocated to the P site. So now it's in the P site. Something else that happens is the mRNA gets moved through the ribosome. Okay? And so now we're in this situation. We've got the mRNA lining up to the P site with the next codon. We've got the loaded tRNA here on the P site ready to receive another bond. We've got an empty A site ready for another tRNA to come in. And the E site, which previously held that one uncharged TNA, is now empty. And this process repeats. And then repeats again. And then again. Let's see what I skipped. So, talked about this. Talked about that. Mentioned those things. Here's that reaction. Nucleophilic attack. Translocation. Translocation is a step that we said moves the growing peptide from the A site to the P site. Okay. Now everything we know about chain elongation, we figured out using these analogs, these structural analogs. So this is puromycin. Puromycin looks a lot like the 3' end of the amino acid tRNA. And so what they found out was when they used this analog, it would go and it would bind to the P site and to the A site. This is how they discovered what P site and A site were, that they were actually things. When it binds to the P site, nothing happens. It sort of just stops excuse me, stops translation. But when it binds to the A site, there's actually a reaction that happens between this molecule and the growing peptide chain. So they know that the A site is where the amino acid that's going to be added next goes in, and they know that the P site is where the current amino acids in the waiting to be elongated chain are. Anyways, the book goes into a better explanation of how exactly they deduced A and P sites from using this molecule. But again, it's a structurally similar molecule, so the reactions were very similar. Chain termination happens when we get to one of those 3 codons that doesn't code for an amino acid. So there's a few stop signals. We've got 3 of them: UAA, UAG, UGA. We're going to need some other proteins here, some release factors. Now there is release factor 1, release factor 2, and release factor 3. Now release factor 1 binds to UAA and UAG. Release factor 2 binds to UAA and UGA. And so they both recognize different stop signals. Now RF3 doesn't bind to a termination codon, but it again facilitates the activity of these other 2. Now there aren't tRNAs that bind to these anticodons, or these codons, sorry, stop codons, but there are these proteins that bind there. And some of what these proteins do is they basically stop tRNAs from being able to come in there. And some of them actually, they change the activity of the enzyme, of the peptidyl transferase. And so their presence causes the peptidyl transferase to hydrolyze that last bond there between the tRNA and the peptide chain. So basically freeing it from that whole complex. And you can kind of see that all happening here. So we get to a stop codon, UAA. These release factors come in. We see the chain gets cleaved off. So again, their presence causes the transferase to hydrolyze that bond. And then they also help with the dissociation of these complexes. We didn't talk about what RF was. I think this is another release factor. RF, unless it's a typo. Okay. So yeah, these are all the components that are required for the different steps in this translation process in prokaryotes. The activation step, and the initiation, elongation, and termination. Now for the most part, eukaryotes follow a lot of the same trend. We're going to call everything a little different. Our ribosomal subunits are a little differently sized. But for the most part, it's going to follow a lot of this. Now the main difference is going to be in this chain initiation step for eukaryotes. There's like 13 additional proteins, initiation factors. And that's kind of the major difference. It doesn't have quite the same thing as a Shine-Delgarno sequence, but then also the mRNA is actually different for eukaryotes, right? It's got that 5' cap and the 3' polyadenylate tail. So we'll have to talk about that. 21st amino acid, selenocysteine. So there are other amino acids, we've talked about this, amino acids that are not found in proteins, but there are a couple others that are found in proteins, like hydroxyproline, and obviously N-formyl methionine. These guys get post-translation modifications, so once they've been incorporated into the, well, for the case of hydroxyproline, Once it's incorporated into the protein, it gets modified. N-formyl methionine we saw gets modified on the tRNA prior to being put into that first position. And selenocysteine is one that also gets synthesized on the tRNA. This one's a little neat. Let's see, do I have another slide here? I don't think I have another slide. Yeah, no, okay. So what actually happens with this one, it's a normally a cysteine, oh sorry, yeah, a cysteine would have an OH here. And so instead of having an OH, it's got a selenium. So the selenium has replaced the oxygen. So this, what normally happens is that, so the other weird thing about this is that this guy does have a tRNA, but it doesn't have a tRNA synthetase. So nothing that will actually put it on, put this amino acid onto a tRNA. It does have its own tRNA, and the reason it needs its own tRNA is because this tRNA recognizes a stop codon. So so this thing normally it recognizes a stop codon. So in any protein where it requires a selenocysteine, we get to the stop codon, and then this tRNA will go and find there. Now, before that can happen, though, there needs to be a misacylation, a mistake essentially, where the synthase for cysteine will mistakenly put a cysteine onto this tRNA. So the tRNA that recognizes selenocysteine accidentally, well, it's not accidentally, it's on purpose, right? But it's a mistake that happens. I guess an intentional mistake. I don't know if you can actually call it a mistake, but it's a mistake because the tRNA synthase is meant to do a really good job of only putting its amino acid onto its corresponding tRNA, and it doesn't in this case. It puts it onto this other tRNA that recognizes a stop codon. You can see how that might be bad. But because this is supposed to happen, when this thing recognizes the stop codon, and it has this selenocysteine or this cysteine residue on it, little differences in that tRNA actually tell the kind of the ribosome that this isn't an actual stop, and so then it just keeps going and it incorporates this in there. Once it's incorporated in, another reaction happens that actually puts in the selenium atom. So 21st amino acid, because this guy shows up, and again, it has its own tRNA, it's weird that it doesn't have its own synthetase. So this is a structure of the ribosome when X-ray crystallography actually got really good, and the purpose of this is to show that the active site of the transferase, which is the protein portion of the ribosome, is actually far from the peptide bond that forms in the growing chain. And so what this elucidated for everybody that was studying ribosomes was that the actual RNA portion of the ribosome is catalyzing this reaction. So this goes and lends further evidence to the idea that in the beginning, before there were proteins, RNAs were actually doing not just storing the information, the genetic code, but also catalyzing reactions. Because if you think about it, right, proteins are required for translation, but if you didn't have any proteins yet, then how did translation happen? Like, how do you make a protein if you don't already have proteins, right? Chicken and the egg kind of thing. So ribosomes obviously were doing the job. Let's talk about what a polyribosome is. So several different ribosomes can all be bound to the same RNA. This happens because in prokaryotes, everything's just floating around in the same space. And so while a gene is getting transcribed, so the mRNA is starting to like kind of dangle off of the actual DNA, ribosomes just go and jump on it and start kind of making their proteins right there, one after another, sort of chasing each other down the mRNA. And this is, again, because we don't have, or because they don't have a nucleus that separates the ribosomes essentially from this replication or this mRNA transcription that's happening. So what you're seeing here is sort of meant to be a big long section of an mRNA, and then there's 1, 2, 3 ribosomes all running along, one clearly having gotten much more protein transcribed than this one and then this one. Your book has a really good picture, and it's obviously not here. Let me show you, since I can just jump over there real quick. Those are my kids. It's a really neat picture of the, it's a real picture of an mRNA, so you can kind of see it. So the mRNA, I think, is kind of like this bit, and these are going to be different protein or peptides that are kind of growing off of that mRNA. So different ribosomes running along, kind of doing that. All right. Let's see. Eukaryotic translation. So I mentioned several differences. We're going to have a 5' cap and a 3' polyadenylate tail. So this gets added to the mRNA after the mRNA gets spliced out of all its introns, right? Spliced all the exons together. So this bit gets added on. And so this is important for initiation and translation. So main difference, eukaryotic translation has a lot more initiation factors. We're going to see there's a few different steps, just like there was in prokaryotic initiation. We've got to assemble the 43S pre-initiation complex. We've got to recruit the mRNA. And this is where the 5' cap helps that. It orients the ribosome to the correct start codon. Let's see. Via a scanning mechanism. So we're going to take a look at that. And this is, of course, ATP driven. And then step 3 is going to be recruitment of the 60S ribosome, forming the 80S initiation complex. So again, going to hydrolyze GTP, and all the initiation factors get released. Let's see if we can see a picture here. So again, the 5' cap, seen here, helps with all of these initiation factors. We've got initiation factor E4, initiation factor F3, or 3, initiation factor 4A. You can see here how the polytail also fits in, right? The polytail has to help with this linking protein to help get all of these other proteins together so that all of this can help guide the ribosome to the correct spot so that it can start at the right spot. Here you see those many other initiation factors and sort of which subunit they're in and kind of what job they facilitate. 3 stages in the initiation. So these are those stages. Step 1 is forming that small complex. Step 2, we just said, was, what did we say step 2 was? Man, my short-term memory is not that great. Orients the ribosome to the correct codon. Recruitment of the mRNA. I guess we could have seen that. Right here's the mRNA, and now it's bound. And then initiation complex 80S is formed. Initiation factors fall off. So then it's ready. Now for the rest of that, it's basically the same. Uses the same mechanism of peptidyl transferase, ribosome translocation. This site doesn't have an exit site. It only has an A site, an acylation site, and a peptidyl site. It does have a few elongation factors. So one that's very much like the EFTU, which is all about making sure the correct amino acids go into the right spot. And then TS, which is all about regenerating TU. And then EF2 is the thing that's most like EFG, which was our translocator. This moved the growing peptide chain from the A site to the P site, if you remember that. All right, chain termination happens again when we encounter a stop codon. No tRNAs recognize this. Release factors will bind to these codons. Different than the prokaryote version, we only have one release factor, and it recognizes all 3, as opposed to, what was it, release factor 1 recognizes 2 codons, release factor 2 recognizes the other 2 codons, or 2 different codons, or one same codon and then a different codon, you know what I mean, and then the release factor 3 didn't bind any of the codons. And then it also, in this case, catalyzes that hydrolysis between the amino acids and the tRNA. Suppressor tRNAs actually let translation continue through a stop codon. So sometimes if mutations to the genetic code switch a letter, probably not the wobble position, but switch one of the other one or two positions, and actually makes a stop codon where there shouldn't have been a stop codon, there are tRNAs that will kind of recognize that and allow translation to happen. I'm not sure exactly what the criteria are for that. Maybe if the peptide chain isn't long enough, or if there's there's obviously a mechanism that's checking to see something about a stop codon to make sure it's a legit stop codon or not. But so that's what suppressor tRNAs do. So we know that if we didn't have a nuclear envelope, that translation would happen in the nucleus, just like like it does in prokaryotes. So that suggests that it was separated on purpose. So there was a reason why we enclosed our DNA off from everything else. I don't know what that reason is, but that would be the logical conclusion to draw, and not just to say, oh, in some life, translation happened instantly during transcription, even. But in eukaryotes, that's not the same thing. Evidence shows and lab results show that if we didn't have that separation, there's nothing stopping translation from happening right there in the nucleus. AUG is not always the start codon. There are also peptides that get synthesized on the surface of major histocompatibility complexes, and they have a different start codon. Or sorry, they're not synthesized on the surface of the major histocompatibility complex, but they are synthesized to be presented on the surface of. And then they have, like I said, a different start codon. So not totally universal in that UAG is the start codon. Post-translational modifications happen. We know that that hydroxyproline gets formed. We know that there's a lot of hydroxylations actually that happen to amino acids, and methylations as well. So newly synthesized polypeptides frequently modified before they get to their final location. The N-formyl methionine is always cleaved in prokaryotes. It's just the starting amino acid, but it's often not even involved or included in the final protein, so it gets cleaved off. Specific bonds and precursors get hydrolyzed, so we know that we looked at that exact example actually for chymotrypsin. We know that chymotrypsin starts off as chymotrypsinogen, and then after some cleavages, it becomes an active enzyme. Same thing kind of happens with an insulin precursor. The leader sequences get removed, and finished protein enters the Golgi apparatus, which will direct it to its final location in the body. We can also get associations made. So what are they called? They're not called complex enzymes. They're called. Anyways, there's a name when a protein gets a prosthetic group added to it, like hemoglobin gets the heme groups. So I guess anything as like a cofactor, disulfide bonds, covalent modifications, additions of lipids or sugar groups to make the final protein. All of these things can happen to the new protein. So these are other just types of modifications, post-translational modifications. Methylations, phosphorylations, arginilations, ubiquitinations. Yeah. And all of these would be, depending on what part of the body they're going in, whether these are going to be proteins on the surface of a cell, whether these are going to be enzymes, whether these are going to be transport molecules, whether they're going to be storage molecules. All of these would have slightly different modifications to them to facilitate their new task. So you can see a couple here. This one is precursor of proinsulin, pre-proinsulin, and it's going to get those disulfide bonds formed, and then we're going to get some cleavage in here at some point. We're going to get some cleavage there, cleavage there. So just because a protein gets translated does not mean that it's anywhere near being functional. Proteins are a dynamic state when they get turned over. Abnormal proteins that are formed and errors in transcription or translation get degraded quickly. You would hope that. Proteins get turned over often. That's what this first bullet point is saying, that the proteins in your body are not there for a long time. They get made, they do their job, and then they get broken down because you're constantly making new ones. The pathways are restricted to subcellular organelles such as lysosomes. So lysosomes and proteasomes for bigger structures are what we use to kind of digest and to break down proteins. Some special sequences, like whether or not, depending on what amino acid you actually have on your N-terminus, can determine how long of a life you have, what your half-life is. So specific signals will direct proteins to a lysosome. A lysosome just destroys anything that goes in there. Ubiquitinylation. This is the process of binding the polypeptide ubiquitin to a protein to mark it for degradation. So this is a post-translational modification that I mentioned a few slides ago. This is a signal to your body that, hey, this is a protein that needs to be broken down. I was mentioning the N-terminus actually determines how viable or how long living your protein will be. And so you can see this here. Some amino acids are actually resistant to this breakdown or breakdown via this ubiquitinylation. Others, like these guys, have short half-lives. So if you have a protein that you want not to live very long, then you go ahead and you do that. There are some acidic amino acids that actually require a tRNA in order to get destroyed or to get sent down the path to destruction. And I think we have an example of that in the book. We do. Let's see. Oops. Your book had a picture. I think that's all I want to show you. It's just the picture that your book had. Unless this is the same picture that I was looking at just a second ago. No, I think this is it right here. So this is a protein that actually has an aspartate. At the N-terminus. So this is a acidic amino acid. And so what happens here, before this thing can be marked for protein degradation, we need to bring in here and change that N-terminal amino acid to be something else. And so it could be a glutamate or an aspartate. This tRNA comes in, brings in an arginine. The arginine now is the N-terminus. And then now that it has an arginine N-terminus, this thing can be marked for degradation. I don't know why this didn't have that picture in it. Okay. So here's a diagram of the degradation by the ubiquitin. So this substrate here is the protein to be digested, ubiquitin added to it, and then this helps the proteasome recognize it. Proteasome is going to go and break this protein down. And then the ubiquitin gets recycled. Oh, here's that picture. Nice. Didn't have to jump around. Oh well, you guys don't mind. And I think this is the last slide. So chaperones and protein degradation process. So misfolded proteins are often handled by chaperone proteins. And I think we might have talked about chaperones before. I don't know if this is the first time I'm mentioning that term. So chaperones help proteins fold properly. or sometimes are used to keep a protein from folding incorrectly until it's in the right region or the right place. And so, what are we looking at here? We're looking how the ubiquination system can take a ubiquinated protein, take it to the protein-bound proteasome. So this was what we just looked at in the previous slide or two. So we get a degraded protein. Now, this can happen to unfolded or misfolded proteins, right? Because in addition to proteins that are useful and that are working and that are not misfolded, that also get broken down, mistakes do happen. And so two options can happen for your broken protein. One, break it down using the ubiquination system and reuse those amino acids. Two, have chaperones that go and actually fix it. And so this is an unfolded protein that gets associated with this chaperone protein, and then using some ATP energy, it gets remodeled, refolded. You can imagine that chaperones are going to be protein specific. One chaperone can't go and tell or help every different kind of protein how to fold. So this chaperone here is going to be specific for this protein and made simply so that if this protein misfolds, they can help it fold again. Because that might be, right, this costs 1 ATP. Remember how much GTP we were costing just every time we brought an amino acid in to try to make a protein? So this is a much more energy efficient way of getting back your good protein that was misfolded. All right, I think that that just about does it for this chapter on protein synthesis. So I think we're going to skip the next chapter, maybe even the next couple of chapters. I think we jumped to like chapter 16 or something after this. I'll have to double check that. Don't quote me on that. But enjoy your spring break, guys. Hope you're doing well. And I will post another video up. Just maybe not as soon as I wanted to, but over the next week you'll see another video pop up, and then we'll be talking about that in the weeks to come. Enjoy your spring break, and try not to be stir crazy there at your house. Hopefully you guys are doing well. All right, see you.